the states. So how to deliver for better way and country become self-sufficient in edible oil production and availability in the country. And as we all know, groundnut is a major oilseed crop with highest productivity and oil recovery. So there is a lot of expectation of the policy makers of this country from this groundnut crop, groundnut researchers, groundnut institute, and ACRI to satisfy the demand of this edible oil of this country. So with this brief background, we will be lucky to listen Dr. Nadaf. Is a renowned groundnut breeder, researcher of this country as well as US Dharwad. Sir, I welcome you wholeheartedly on behalf of my institute and entire ECRIP group. Hope will enrich our idea, research, and all these new breeders, new scientists, freshly joined all scientists from ACRIP, they will also will be benefited from your lecture, idea, and your experience. Thank you, sir.
Thank you, Virat sir, for your opening remarks. And I will take only a few minutes to briefly introduce you today's speaker. Dr. Nadaf sir did his undergraduation from US Bangalore in 1980, followed by master's in 1982 and a PhD from US Dharwad in 1993. Later on, he joined as an instructor at US Bangalore, and then he moved. A couple of time he spent with the agriculture officer as a Bank of Maharashtra in Pune, and then he joined ACRP and as a scientist groundnut at US Bangalore, and later he was selected as senior scientist at US Dharwad under ACRP groundnut scheme, later promoted to principal scientist groundnut. And later he headed the scheme and as assistant director of research as well as director of research. He retired as director of research from US Dharwad. And he has, during his career, he has guided around 15 MSc students and 10 PhD students, and more than 83 research papers were published from Dr. Nadaf group, and he has handled around 21 projects and mobilized around 4,176 lakhs during his entire tenure. And he has received prestigious awards from Biotechnology Overseas Association Award in 97 and Outstanding Contribution in Seed Production and Seed Research Award in 2005 and Best Dust Testing Center Award in 2011. And sir has developed a couple of varieties, DH40, DSG1, DH86, DH101, and DH4-3, and identified tag 24 and collaborated in developing mutant 3 entry. And he has visited several countries, USA, Netherlands, Spain, and during his tenure. And for more about his publications, you may visit ResearchGate link that has been provided. This slide will be shared in the group. Thank you. And with that, sir, I request Nadaf sir to present today's talk, sir. Thank you, sir.
Thank you very much, both Dr. Bera, Director DCR, and Nataras for my introduction. And I am very happy to be associated with this initial lecture on successful groundnut breeding. Now, this idea of involving the retired scientists or workers is a good idea because many people have their own experiences, and these experiences will help the younger generation to gain some insight into their experience, and maybe if they are useful, they can use in their research work, and it can help them. But in my case, actually, I am going to talk about only the practical tips. I am not going to give the theoretical background. It will be mostly the practical tips, how we can actually achieve or be successful in groundnut breeding. Now, first of all, we must thank God that we have been given the intellectual capacity, because other creations of the God do not have this, but we do have the intellectual capacity. We must use that for the betterment of the society. Now, the task given as a breeder or as a scientist for us is how we can actually help the farmers who are actually innocent people. They are waiting for our research results or research products which can help them to elevate them from the poor situation. So that is why we must thank God first for giving us the intellectual capacity, and we must therefore work sincerely to the best capability of us to give the best result. That is, in my opinion, we must try to be sincere, honest, and keen in our duty in whichever place you are working. We must be sincere and honest in working. Now, let me tell you that we are only trying to utilize whatever the natural variation that has been given in nature. Say, for example, the germplasm that we have at the nation or even at international level. Now, that natural repository of the natural variation, we should use it properly for developing the variety or any research that helps the farmer to get the best. That means to get either higher yield or higher quality of the product. So we must try to combine the genes from different germplasm to develop a variety or a product that is useful to the farmer. Now here I would like to stress on the ethics part, because ethics is more important, because unless we are very sincere and honest in reporting whatever the results that we get properly, our efforts while actually compiling the data and presenting it for further advancement will be futile exercise if there is any deviation from this. Therefore, the performance of the genotype can be pulled down by a single person if he manipulates the data in any way. That should not happen. We should actually report the data as it is, and if there is any deviation from the normal, it should be explained why it has happened, or for any lacuna or any deviation in the data, for example, could be because of poor germination of a particular genotype, or could be, let me give you some examples where we used to, as a, although we were working on groundnut, but Sysamum was also given to this center as a voluntary center for testing. And many of the or many of the entries that were received for testing here in either initial varietal trial or advanced varietal trial, Dharwad, it is a transitional zone, receives very high rainfall during the Kharif season. And the genotypes that we were getting were highly susceptible to both leaf blight as well as Cercospora leaf spot. Because of these two diseases, majority of the actually genotypes, even they failed to flower also. So we could not get anything. But those which were developed here, naturally they have to survive and yield something. So they were better. It doesn't mean that we manipulated the data, but the real situation should be explained properly so that we get the real data on our hand. So whatever the genotype that suits to that particular geographical location should be promoted. So in this way, we should actually give the proper reasoning for if there is any variation or deviation from the normal data. That is one of the uh, thing that I wanted to stress. And let me tell you, each location actually, or each geographical location, wherever you are located for doing the research, it will definitely have the unique features. We should try to characterize it first. Say, for example, the soil characters or environmental characters. We should thoroughly characterize it, and also you should get acquainted with what are the features of this, or what are the requirements of this particular location to grow a crop successfully. For example, groundnut, which features are required. And uh, in this manner, you can actually try to identify what are the objectives, your objectives of either breeding or agronomy or for entomology or pathology, whatever. But your objective should be such that they should be farmer oriented. We should not actually do the research as per our wish, but it should be farmer oriented, because the priority should be given to the the farmer needs, local needs, and we should identify the most limiting factors. Which factors are actually decreasing the yield level, potential yield level of the groundnut in that particular area. Now, we we also uh, try to do this in two ways. Actually, one can do the farmer survey and try to contact as many farmers as possible in your location and try to get the feedback from them. That is one way. And most of the farmers, they will explain their problems to you, and you can note down which is the common problem that is shared by the farmers. And this can be actually verified by doing the experiments on your farm. Whether a particular, say, disease or the pest or any particular, say, the nutrient management is a problem, or water or the rainfall is the problem. You must try to experiment and try to find out which is the most limiting factor, and the next limiting factor, and the next limiting factor. In this way, you can actually identify the problems. It should be not only to your research center, but it should actually represent the entire area for which you are working. Uh, that is possible by experimentation. Uh, it may not be at your single location, but it could be at your target locations also, so that you will clearly come out with the most limiting factors of groundnut in your area. So once you do that, you will come to know, say, for example, in our location, that is Dharwad, disease pressure will be very high during Kharif season. That is the most limiting factor, of course, in only the transitional zone. If you move to the north of the Karnataka, there it is a dry area, there it is not a problem. Disease is not a problem, but there the lack of moisture during critical stage is a major problem. So it differs from location to location. So within the state also, there will be different agroclimatic zones. Each zone has a specific problem. You must try to identify that, and based on the area of coverage of groundnut in that particular location, you must prioritize your research. Now, another thing is the extension people who are working either in your university or the ICR system, they will also come to, because they interact with the farmers more frequently than you. Therefore, they will give you the correct picture of what are the requirements of the farmers. So their opinion also should be taken into consideration. And then you can also discuss these issues with your central team. Say, for example, your PC unit, they will also be giving you the objectives of the research, and it is usually based on the local needs. You can discuss them and finalize the objectives of your research. So this way you can fine tune your research objectives so that nothing or the expenditure that is made on research should not go waste. Your target should be the farmers first. Now, apart from this, actually, you must have the constant touch with the farmers. Some of the farmers are very innovative, and they will be doing much better job, and they will know the things. Say, for example, I think BRC people as well as we came across with, say, there are many farmers who are very innovative, like Jai Kumar Gunde from Pune. Actually, he has more than 10-acre land, but he actually adopts a very good BBF method with polythene mulching, and he does very well the seed production. And many such farmers are there. Even in Karnataka, we have some farmers who are very innovative, and they grow a lot of groundnut. And every year they will be successful. And we must be in contact with them.
ನಾವು ಇಲ್ಲಿಂದ ಶೇರ್ ಮಾಡ್ತೀವಿ ಅಂತ ಹೇಳಿದ್ರು ಸರ್. ಅವ್ರು ಅವನ್ನ ಇಟ್ಕೊಂಡು ಕೂತಿದ್ದಾರೆ. ಪಾಪ ನಿಮ್ಗೆಲ್ಲ
Now, I was telling you about the contact with the farmers. We must establish the strong relationship with the local farmers, and they will be helpful in many ways. I am going to give you some of our experience with the farmers in the next. Say, for example, one of our target area was actually the residual moisture groundnut area in Ankola, that is coastal Karnataka. There we had one farmer called Paide. He was actually a veterinary doctor, but he had a land of about 5 acres. He used to give at least 1 acre for us for experimentation. We used to conduct research on his farm. Sorry?
Words.
Yeah. So, if we have a very strong relations with some of the farmers, or they will help you in many ways, and we can get very good results. Now, coming to groundnut crop as such, from the breeding point of view, it has many challenges, and it has also unique features. We must try to understand that. Of course, we know that the flower structure is because of its clistogamous nature, it is highly self-pollinated, because of which we have less genetic variation. And you are aware that even with the huge germplasm that we have in cultivated groundnut, the amount of genetic variation is less compared to other crops. That is true. But on the other hand, the diploid wild species, they have much variation. At the genomic level, it has been proved that cultivated groundnuts have less variation compared to the wild species. Although limited in number, they have more variation. But still, if you are able to alter few genes in the cultivated groundnut, it can produce lot of variation. This is also proven. If you can alter few genes, you can get lot of variation in the cultivated groundnut. That is the plus point. So you can play with, or you can try to breed the varieties with limited, even with limited genetic variation. Another problem in groundnut is it has very low seed multiplication ratio, and also the seed rate per acre is very high. These two features actually again limit the spread of the new variety to a large extent. And also the number of seeds that we get per cross is also very less compared to other crops. For example, if you make the cross in other... Use this one. Next. So here number of seeds, crossed seed we get is very less. But still, you can try to make the crosses. And here my suggestion is you should not attempt more crosses and produce less number of seeds per cross. That will be a futile exercise. But instead, you try to select the crosses which are best for you and produce more crossed seed per cross, so that you get larger population, and then the selection would be possible. Otherwise, if you make only few hybridization per cross, and you may not end up with any success in that. That is why you must try to attempt more number of hybridization per cross. Limit your number of crosses, but make more hybridization or more crosses, get more seeds, and then it is possible to make the success. And another problem is, unlike other crops, you cannot see the economic part of the crop because it is below the ground. So you cannot actually, until you uproot it, you cannot see the results. This is another problem. But here, by experience, you can try to know the yield of a plant based on your experience. It is possible to guess, or of course, sometimes you may select a particular plant for high yield, but it may be turn out to be poor yielder when you uproot it. That is quite possible. But based on the parents involved, as well as the conditions, you can say, for example, if you have involved the high harvest index parent in the univer cross, then even with the low growth also you can get a higher yield plant, high yield plant, that is possible. But still, unlike other crops, you need more or better experience to make the selections in groundnut. We will come to later, we'll discuss about that in a few minutes. But quick loss of seed viability is another problem in groundnut, because you cannot keep the seeds for long time for future use. You have to grow it every time, or every at least once in a year during Kharif season. But if you grow it again in summer season, it has to be again grown in Kharif season, because seeds cannot be carried forward. This is another limitation, but if we have cold storage or other facilities, it will be useful. But many a times this is another drawback of this crop. Now, groundnut also gives us many unique opportunities, because it has many health benefits. Actually, from my childhood, I love groundnut, and in fact, God gave me the opportunity to work on this crop. I have seen in many of our North Karnataka area, along with the food, they give a handful of actually groundnut seeds for eating. Actually, if you eat a handful of groundnut seeds, you are healthy, not only healthy, but also you feel that your stomach is full. And that actually, it gives many nutritional benefits also. And this unlike other oilseed crops where they can only be crushed for oil, but here groundnut crop can be used both as a food as well as for oil extraction. That is the advantage. And I hope in future actually groundnut should be promoted as a healthy food compared to the crushing for oil. In Western countries, it is already being done. Their majority of the produce goes only for healthy food, but less, because alternatives are there for oil extraction. But for healthy food, this is a very good crop. One should actually explore this as a healthy food. In many of the nutritional features, like minerals as well as vitamins, apart from the high oleic acid, actually it increases the not only the shelf life, but also it is healthy from the point of view of the heart and other diseases also. So as a healthy food, it should be promoted. Now, that is why I said it is groundnut is almond of the poor, but not only poor, even rich people, they can consume it and be healthy. That is possible. So specialized foods in future, I hope they will be common with the groundnut. So actually, this gives us the scope for groundnut improvement as a healthy food, as well as even some portion of it can go for oil extraction as well. Now, coming to actually the, of course, breeding as such, I feel that every breeder should have his own breeding material. We should not depend on anybody. This is my opinion. We should have our own breeding material, because our problems and our local needs and our local adaptability is quite different from others. So therefore, we should try to develop our own breeding material. We should not depend on others. And you have already prioritized your breeding objectives. Then you can make the selected few crosses, as I said earlier, to produce or more crossed seeds per cross, and then raise the large F2 population in the target area. Now, another problem in groundnut is, as I said, low multiplication ratio is one of the problems. Now, in this case, if you are able to grow the F1 in a very high fertile, well-managed conditions, you can get more seeds per plant. That is possible. I remember one farmer in Gadag area, when we were actually we developed the DH256. Our seed quantity was very handful, but we gave, fortunately, we had given to farmers for on-farm testing. Hardly we had given 2.5 kg to one of the farmer in near Gadag. He had wisely grown in actually very well-maintained and drip conditions and organic well-maintained soil. And from 2.5 kg, he had multiplied it to more than a quintal because of that well-maintained conditions. And he had shown us the number of pods were more than 100 per plant. Now, this kind of the well-maintained conditions to get more seeds per plant can be done at each of the research centers, so that the breeder can actually grow the F1 in such conditions and try to increase the seed number so that he can get large F2 population for selection in the next generation. That is possible. Now, here large population is required because yield as well as its component traits, including the quality traits, except a few like oleic acid or others, although they are mostly the quantitative in nature or polygenic. And as the number of genes involved in contributing to the trait increases, you have to grow large population. So large population is quite essential. So we have to, these are the possible avenues to increase the size of the F2 population. Now, first thing is you must limit your number of crosses, and you must try to grow F1 in very well-maintained, fertile and conditions, so that you can get more number of seeds per plant. Now, of course, once you produce large population, now the next thing is selection. Of course, we are going to discuss about that, but if you, of course, this sufficient variation, of course, if there is no sufficient variation in the existing germplasm, of course, we have to go for other alternatives like induced mutations or others. But sometimes you can use hybridization followed by mutation to increase the variation. Sometimes if the traits are distributed in multiple parents, then you can use multi-crosses also to increase the variation. So the major focus should be to develop your own breeding material so that you can select from that the varieties that are required for your local location. Sometimes these varieties developed could also perform in other locations as well. but your focus should be for your target area. Now, the selection process is very tricky in the sense, as a breeder, you must actually try to understand the crop first. That means you must try to learn how the plant grows from germination till harvest. That means you must have keen observations of the plant, how it grows, what are the periodical changes that takes place over the period of time. It could be in seedling stage, vegetative, reproductive stage, seed fillings, as well as maturity. Now, if you are keen to know, say, for example, your early vigor, you should be very keen in observing the plants at the seedling stage itself. Sometimes we used to go to the field after the sowing, and some of the entries germinate very quickly and grow very fast, but some others, they take a lot of time to come out of the ground and also to reach a certain stage. Now, this kind of early vigor is also important in many cases. So your keen interest is very important. Keen observation is very important to get the desired results. And also vegetative stage, how much time it takes to pass the vegetative stage, and the reproductive stage, how much period it is there in the reproductive stage, in the sense flowering duration, how much it produces, and how it actually converts into the pods, and maturity wise it is early or late or whatever it is. So you must be a constant observer in the field. So then only it is possible to be successful. Otherwise, if you go to the field once in a while and observe, for say, for example, the PC unit has given the list of characters, and if you go only for recording those, it will not be useful. It may not be a successful breeding. Therefore, you must try to visit the field as frequently as possible. I say in the morning hours, it would be very pleasant to visit the field. First, up to lunch, your field visit should be the priority. And in the afternoon, you can design some of the experiments to confirm whatever you observe in the field, whether it is actually true or it is only a random phenomena. You can design some experiments and try to prove that whatever I observe in the field is actually true. You can try to design some experiments and prove that. That is also possible. Now, as I said, selection, it all depends on how keenly you observe. Say, for example, if it is a drought resistance or drought tolerance, unless you have the drought situation, you cannot select for the drought resistance. That is simple. But if we have a severe drought, let me give you the example of our case of identifying both the DH256 and 257. We had made the cross, and we had grown the F3 progeny rows, right? The field that you saw here in the first slide. There we had grown the F3 progeny rows, and that year was very severe drought condition. And we could see some of the progenies were still green, but while others actually which were susceptible, they were already collapsed. That gave us the strong evidence that these could be the genotypes which are actually drought tolerant. And immediately, this was the Kharif season, and immediately the next season we took them to the summer, and we actually tested them both under well irrigated and limited irrigation condition. And we proved that the checks varieties actually, they suffered because of lack of moisture, but these genotypes actually perform better even under limited moisture conditions. So this kind of your alertness and further testing is very much required. Then only you will be successful. Now, similarly, if I say, for example, disease resistance, if there is a severe disease incidence, then only you can select. Otherwise, everything will be same. You cannot, if you go to the dry area where the foliar diseases are not there, you cannot select actually for foliar diseases in dry area. But if you come to the Dharwad or Vruddhachalam or some centers where foliar disease is a quite, or these are hotspots, there you can select for disease resistance. So similarly, based on your objective, your selection pressure should be proper. So unless you get the condition for that, your selection may not be useful. Therefore, you should try to select under optimum conditions and then confirm their superiority by further testing. Say, if it is for disease, test them under high disease pressure condition. If it is for yield, of course, you can test them in randomized or RBD or other trials, other designs, and then prove that they are much better compared to your check varieties. And always try to multiply the seeds during off season or during the say summer season or whichever season you get. You must try to always multiply the seeds with under good conditions. Now, in this case, if you find that the genetic gain that we are getting is quite insignificant or less compared to the target that we had, in that case, what you can do is, now here it is quite possible that some of the component traits, say for example, drought tolerance, we actually tried to explore even the root traits under severe drought conditions. If they are faring better, what could be the reason? Always, as a scientist, we must be asking the reason why this is happening. You must be keen to know or answer this question. Unless you develop that kind of attitude, our scientific temperament will not develop. So therefore, we must always try to ask the question why this is happening and try to answer that by designing very small experiments. It is quite possible. Now, I was telling you, if the genetic gain or whatever the expectations you had, if you do not reach that, or if a particular cross is not giving you sufficient genetic gain, in that case, as I said, some of the component traits could be distributed in different parents or different, say, for example, your selections that you made. In that case, you can try to recombine them again by crossing in the early generation, by selecting the genotypes in the early generation and again cross them so that you can recombine them together so that you can get the better genotype. That is possible. This is one way of improving further or getting the better success. I was explaining you about we had for drought tolerance, actually, we tried to explore the root characters also and some leaf characters, why they maintain high relative water content even under drought conditions, whether the number of stomata is less or transpiration rate is low. We try to explore those cases also. So like where you can try to explore the different component traits that may be distributed in different parents or genotypes in early generation. Now, in some cases, there could be some, as I said, if you are selecting for disease resistance, if there is no disease, or if your selection process is for drought tolerance, if there is no drought, under such circumstances, you can go for, you have to retain the whatever the genetic variability that you have, it should be retained, and you can follow the different methods like either SSD or modified single seed descent method, or in some cases the bulk method. If you have very limited material, you can even follow the bulk method, but which is more scientific as well as confident, you should try to adopt that, because we cannot actually advocate the same method in every conditions. It all depends on the context under which, or the situation under which you are working. We cannot recommend a particular method for everybody. That is not possible. So in the under low selection, or if your selection pressure cannot be actually say, if there is no disease or no drought condition or some such situation occurs, then if there are proven molecular markers, they can be used. That is quite possible. Otherwise, you can use the SSD or other methods. Now, there are some traits which are highly sensitive to the environmental conditions. So there, whatever the selection that you make, it may not be the true representation of the gene or genetic architecture. It could be, majority of it could be only environment. Under such circumstances, you must try to increase the number of locations for testing. So if you increase the number of locations for testing, there you are going to reduce the environmental influence, and thereby your selection will be mainly based on the genetic architecture of the genotypes. So that way you can reduce the environmental influence. Now, another important thing that I would like to stress is every season, for that matter, every season of evaluation has its own unique features. Say, for example, a particular season has severe drought and you cannot actually get the yield of a majority of the genotypes, but still you can use that situation very efficiently to screen all your germplasm, all your breeding material, all your even released varieties, whatever you have grown in the field, it should be thoroughly screened for drought resistance during that year. Now, if during the particular year, if there is a heavy rainfall at the time of harvest, and it has become very difficult to harvest your crop, under such situation also, you can screen your material, entire material, for fresh seed dormancy, as well as those which can withstand that kind of severe or high rainfall situation. So what I mean to say is every season should be effectively utilized for your selections. Now, that means every season has its own uniqueness. You must characterize that. For example, a particular season you may experience very high temperature during flowering period. Now, that may reduce the seed set or the total seed set percentage or the pod formation. Now, that season also, that means that you must characterize a particular season very effectively both environmental as well as your soil conditions should be characterized perfectly, so that the selection whatever you make during that season will be very fruitful, and you can later design the experiments to prove that whatever selections that you made during that season are very useful. For example, if there is a heavy rainfall and everything gets sprouted in the field, but some genotypes remain intact, that means that they have the dormancy period, and you can test that in the lab also and prove that these are the genotypes which are having very good fresh seed dormancy. Like this, actually, you can try to utilize every season effectively. That is quite possible. Of course, wherever you have the molecular labs, you can use it as a tool. But in my opinion, ultimately, field is the best actually lab for testing the genotypes. So there is no compromise for field evaluation. Field evaluation is first priority, but you can use both lab as well as your other small experiments to prove that whatever that you observe in the field, whether it is uh, correct due to which particular feature, you can actually prove that by simple small experiments. So this we discussed traits. Now, another important thing that I would like to highlight is integrated approach actually gives you much better progress compared to working in isolation. This is this will help you in many ways actually, because a single person cannot master everything. We must depend on others. Every field of specialization has its own advantage or own merits. We must take advantage of that. The AI therapy system is the best. Actually, it gives an integrated approach. We must work together. And in fact, Dr. Chanbhai Gowda and we used to work together. And as a team, we used to go to the field, taking entomologist, pathologist, agronomist, and even sometimes physiologist also from the department used to join us. And we used to move to the field together. And if there is any particular plant of special interest, everybody used to look at that from different angle. And that gives us confidence. Now, if you frequently move with the team, actually you will learn a lot of things. As a breeder, we know only the breeding aspect, but agronomist will tell you which nutrient it is lacking, or some pathologist will tell you which disease this particular plant has and which is not having. Like that, and our actually entomologist R.K. Patil used to work with us together. And although he was an entomologist, he used to spend more time in the breeder's field. That was the time when, because Podoptera was the major problem in our area, and during our close observations, actually, he could identify that the pathogen that actually infects that particular Spodoptera numerarily, he identified, and he again immediately worked on. He took many projects on that and also worked. So it was only because of keen observations as well as collaborative work it was possible. So it is the collaboration or the integrated approach is more important. And once you actually develop a particular variety or a technology, we must share the credit so that nobody is actually denied of any particular technology credit. So, of course, I talked about multiplication of the identified genotypes under high management conditions, so that you can actually give it for further testing. So as I said, ECRIP system is the best system. It is well recognized over the world because you can actually exchange not only the material, genetic material, but also you can exchange ideas, and also you can actually exchange the materials also, and it is for the benefit of both farmers as well as the consumers. It is the best system. We should take advantage of that. As I said, the advanced labs could be used as a tool to hasten the process of the breeding, and it is possible. It increases the accuracy as well as it reduces the time. It is quite possible. But every center will not have this facility. Maybe centralized system or some collaboration can be done. It can help in reducing the time period. So this integrated approach we have already talked about. And lastly, I would like to say that every scientist should develop his own unique specialization, in the sense you could be a breeder, but a breeder can also develop a particular specialization. He should master it, acquire thorough knowledge about that, and he should be an authority over that. That is, in my opinion, every scientist should be recognized for a unique specialization. Unique specialization should be acquired, and you must try to develop material in that. Not just you cannot be a master or authority without the material or without the many publications in that, and also many products out of that. So you must try to acquire uniqueness amongst the scientific field. That is quite possible. If you are young and energetic, it is quite possible that you can acquire this particular specialization and be wherever the knowledge is there, you try to get. Nowadays, it is only the knowledge is at the fingertip. You can get it very quickly, and also you can contribute to the scientific field. So I suggest that for everything that you do, you must try to develop the scientific temperament. So you must plan it very systematically and meticulously. You must implement it, and keep the record of everything, including photographs or videos, live videos of whatever you do, and try to publish them in the highly reputed journals. This will help you in actually your recognition, as well as in recognizing the work done by the DGR at national level, and also you could be recognized at even at international level. It is quite possible. So these are some of the practical things that I wanted to share with you. These may not be the best. You are going to hear from very renowned persons like Chanbere Gowda tomorrow, and several others will be there. You can hear from them also. Try to implement them in your research program so that you can get the benefit out of that. Thank you very much for your patience, and also for your valuable time. Thank you, one and all.
Thank you, sir. Thank you for your presentation. And now we'll take few questions, sir. And I request the participants to raise their virtual hand, and we'll notice them. I'm happy to share that today we have reached the maximum limit of Zoom participant attendance. So many of them have called me and they informed that they are unable to join because the upper limit has reached. So that shows the interest in people to listen to Nadaf sir.
Hello.
Yes, sir. Sheshgiri sir.
Yeah.
Yes, sir.
Yeah. I was closely associated with Dr. M.V.C. Gowda's group, and I know Dr. H.L. Nadaf personally, and I had a chance to work with him also, both on evolutionary aspects as well as the breeding aspects. Hello, sir. How are you?
Yeah, fine. Sheshgiri, how are you?
Yeah, I'm good, sir. And I became an entrepreneur, and I'm very happy to share with you whatever the University of Agriculture Sciences Dharwad has produced the varieties. As an entrepreneur, I have been using them in my business ideas, especially the groundnuts. So, as you said, each breeder should have his own entity, what he wants to work. So, like that, each variety from such prestigious universities and the breeders has given an opportunity to the entrepreneur so that he can develop a cluster of farmers and he will give one particular variety there, start seed production, and then selling the seed, and then making it to commercially happen. And especially we have been working on minimization of the chemical, but we use chemical nutrients, but zero pesticides. And by enriching the soil, we have observed that not only the quantity of the produce, but the quality of the produce is also good. Especially with respect to the storage qualities we have seen. So by utilizing all these aspects, I am making a good business of the university released varieties. I would like to acknowledge here my gratefulness to the
congratulations. Sheshgiri, where you are working now?
I stay in Koppal, sir. This is my headquarter and my native place also. Myself and my wife, Dr. Anuradha, a social scientist, we started our own company, Beyond.
Very good.
Yeah. It is basically on plant and soil health.
Okay. Nice.
So groundnut is one of the important components of our business.
Okay. Yeah. Now, as a food crop, it is gaining importance actually. One can try as you have entered the entrepreneurship. Many people can actually enter in this because nowadays the new products with even high oleic acid they are coming, and they will have increased shelf life as well as the health point of view also they are good. And if the scientist can involve such and produce such products, it is quite possible that it is to increase the value of the produce. Also indirectly we are benefiting the farmers with higher rates for his produce.
Yes, sir. And slowly I am developing our network with the value additions also, the industrialists who are working on groundnut products ready to eat. So in this case, as you said, all these quality parameters, if the breeder and entrepreneur can focus together and share the ideas, so it would be very nice.
Yeah.
Thank you, sir. Thank you. And now I request Ajay sir from our regional station, Anantapur.
Sir, good afternoon, sir. It was nice to hear you after a long time, sir.
Yeah, good afternoon.
I was very fortunate to be a student of both of you and then Dr. M.C. Gowda.
Yeah.
So I learned a lot from you, sir, from both of you. Sir, because I just want to know about the DS256 because you were the earlier you were the trader you were involved in the making selection of those DS256.
Yeah.
Because I was talking to our Yanagi and all, so they were telling that Nadaf sir only he only made the selections, and then it is performing very well even under a drought and also under irrigated. So just can you just brief me about
now the story about the DS256 and 57. Actually, you know that I was actually posted in the seed unit there. There it was only seed production. But when I was posted back to research during the last 5 years of my service, actually, then I was thinking how to break. Actually, you must have noted that the two varieties from ICRISAT, which were actually released by the Raichur Center, they were topping for yield actually. They were the either first or second in every zone. But you know, their pod characters were not good. And also selling percentage was very poor. And they could not be accepted directly for production. So I thought these must be having some good characters. We must improve the pod features of these, and then probably these must be having the drought resistance or moisture limitation. Even under moisture stress conditions, they are performing better. Therefore, we try to involve them in crosses, and we raised as I explained during my talk, we raised the F3 generation. Fortunately, during that year, severe drought occurred. And we could see some of the progenies actually, they stood up even despite the severe stress, but majority of the Czech varieties and others actually, they were almost collapsed. We could not get even one or two seeds, but these actually we could get some good harvest out of this. And also, when we gave these to the farm trials in our location, we went along with our team. We went to one of the area in Bagalkot district actually. There, farmer he had grown both TME2 as well as DS256 side by side. And TME2, there was not a even single seed was there. But in this DS256, there were about 8 to 10 pods were there. So that is how we selected this particular genotype. And we again later, as I said, we subjected this for severe testing under both irrigated as well as limited moisture conditions. And there also it proved better. Now, DH256, the best part of it is that it is suitable for both Kharif as well as Rabi summer season. And it has, under limited moisture conditions, it yields better, but under high moisture conditions also, that means under irrigated conditions also, it produces more branches and more number of pods per plant, thereby it yields better. So that way, these genotypes are much better even under drought conditions.
So, because last this Kharif season we gave it for FLD.
Okay.
So in nearby Anandpur and then surrounding mandals.
Okay.
So under irrigated condition, few farmers they harvested up to 40 tons, 40 quintal also.
Oh.
Under purely irrigated condition, they harvested around 15 to 20 quintal.
Okay. Yeah. It has the potential.
Yes.
Now you can use such genotypes for further improvement. You can develop, because as I said, now if you are looking for drought resistance, there are many ways. Like, for example, this particular genotype, it may be having few features, but as I said, DH257 is another genotype, which has, you must have noted the leaf thickness of that is very high. And also, number of pods is not very high, but if you see the weight of the pods, it is quite high. That means the pod weight per unit volume is quite high in that. So you can try to combine the features from different genotypes. You can still get better genotypes.
Sure, sir.
And in fact, you can talk to Ramesh, but actually he tried to screen these genotypes by his Japanese counterpart, and they have identified some markers or some of the molecular things were identified, and these can be, I think, utilized in breeding program. You
Okay.
can talk to Dr. Ramesh, but
Okay, sure, sir.
we'll give you some insight into it.
Mhm.
Yeah.
Thank you, sir. Thank you, sir. Sir, I have a couple of questions
Okay.
because I am looking into the constitution of AICRP crop improvement trials.
Okay.
Sometimes we face issues with some centers like the supply of seed. Though they have supplied seed to IVT, they are unable to supply the seed to AVT. So, do you have any suggestions for such centers to maintain the steady flow of the entry seed?
No, if they lack the basic facility, for example, irrigation.
Yes.
If they absolutely do not have the irrigation facility, maybe you can assign some seed multiplication to some of the neighboring centers, because if they do not have absolute facility, we cannot force them to produce the seeds. How can they produce if they lack the basic facilities? So you can assign that to other neighboring centers, or that is possible. Only neighboring centers will be beneficial. Otherwise, they can also on their own go to the centers in their state where the irrigation facilities are there, multiply them during the off season, and then give you the seeds during the main season for evaluation.
Right, sir. We'll definitely will take this your suggestion, sir. And sir, one more thing, the number of the germplasm that registered with NPPGR,
Okay.
it's not encouraging in recent days. Not
Okay.
many of scientists are working towards that germplasm registration. Any suggestions from your end, sir?
No, as I said in my talk, actually, every breeder should try to actually develop his own material. Now, once he has developed and identified a specific feature in that particular genotype, he must actually try to publish that particular information. And after thorough testing, now once that is confirmed, then it becomes very easy for registration. There should not be any problem. But there should be some uniqueness in that particular genotype. Uniqueness in the sense, added advantage. Say, for example, earliness is there. Now, he must prove that it is early compared to the existing ones, or it may be an addition to the existing list of earliness. Even that is possible. So it is quite simple. If you have a particular genotypes with a particular trait, and if it is new, you can actually try to publish that and then submit the application along with the seeds to the NPPGR. It is easy for registration. There should not be any problem. And I suggest you to actually, in these days of competition, if you keep some either marks or some encouragement for registration, people may come forward for more registrations.
Right, sir. Right. Sir, now we'll take question from BN Motagi, sir.
Yeah.
Sir. Good afternoon, sir.
Yeah, good afternoon.
So some network issues, sir. That's what the video is not on.
Okay, okay.
Thank you for your nice enlightenment to the groundnut community, sir. So you are our mentor and Dr. Chandbere Goda, sir.
Yeah.
So almost 2 to 3 decades of your together work with the joint effort with the ICAR-IIGR DGR and some of the international institutes. So has helped us to bring in limelight some of the groundnut varieties released from our university under the aegis of DGR. So the present director is nurturing and mentoring the young groundnut breeders also. Thank you for your effort and continued support also, sir. So as Dr. Nadaf sir indicated, so about the DH256 and 257 varieties, how the old R2001-2 and 1 and 2, the highest yielding varieties and their undesirable future has been, defects have been rectified through continued breeding. So a lot of your materials, sir, around 150 plus populations developed at with your effort earlier through SSD method and others we are handling, and with the collaboration with IIBT, Dr. Ramesh Bhatt and his colleagues and many of the students, we could make use these genetic and genomic resources and publish a lot of high impact journal also. So GPBD4 is one of the such success stories, and tomorrow we'll be hearing from another mentor, Dr. MEC Gowda. Thank you very much, Dr. Bera sir, for bringing this important series of the talks across the experience by the different institutes, to begin with US Dharwad. So at present, sir, many of the even PG students across the globe, including West Africa, where I worked with ICRISAT groundnut breeding, they were also joined. One of the Nigerian scientists who worked with Dr. H.L. Nadaf, Emmanuel Sangadwale, so he also joined in between. He's having some issues with this one, and even the presently some Davangere and other traditional university scientists and students who are working on biotic stress also joined, sir. So to make use of this opportunity, so we have developed some crosses between DS256 and 257 also, sir. So a lot of other material, including some aflatoxin tolerance, others, they are in PYT and LYT. Dr. Suma and colleagues at AICRP they are handling and evaluating for pushing them to the AICRP and state trials also, sir. So we are continuing your efforts, sir. Hopefully some couple of varieties will be released and used by the farming community of the region, sir.
Thank you.
Thank you, sir.
Thank you.
Thank you, Motagi, sir. Now we'll take question from Ashok Meena, sir.
Good afternoon, Nadaf sir.
Good afternoon.
And good afternoon, director sir and everyone. Sir, actually I am Dr. Ashok Kumar Meena, ICRP in charge at Rari, Jaipur. And I did my PhD from your university only at US Dharwad under Dr. S.S. Patil, sir.
Okay. Yes.
Sir, my question is, sir, we cannot predict the yield of groundnut plants before harvesting. That is basic, everyone knows. Sir, at the I just working with groundnut from last 2 years. What I have seen at the flowering time, sir, it can help if some plant is giving more flowers at the time of flowering. It is correlated with yield or not, sir? In our observation, we can include that plants which is giving higher flowering at the time of flowering, sir. That is my doubt, sir.
Yeah, it is quite possible, but the thing is, more flowers per plant sometimes it can be converted into more pod yield. But sometimes if the internodal length is quite large, the flowers produced at the higher internodes, they may not actually result into the real pods. They may remain as a peg or at the most some undeveloped pods. So that you must see the traits in such a way that if the flowers are produced in large number at the base of the plant, that means if it is possible for reaching the ground quickly, that means a short distance is there, then this number of flowers produced per plant can give you higher yield. That is possible. But if the plant grows very long, that means internodal length is quite high, and then the flowers produced at higher internodal internodes may not result into the pods. That is the thing. Yeah, sometimes, for example, the varieties like TAG 24, it is known for that. It is a very small plant, but it produces more flowers, and it gives higher yield also. That is true. Yeah.
Okay, sir. Thank you. Sir, one another doubt I have when we are going crossing in groundnut, because I did in our cotton, so it is very easy, but in groundnut it is not that much
Crossing,
easy.
yeah.
Yeah.
Actually, I wanted to talk about that since now there are many actually deviations in crossing technique. Different people use different methods.
Okay,
But at our center, we used very manual crossing. We do not use any forceps or anything. But if you select in the previous evening, actually, you select the bud which is going to open tomorrow.
Okay, sir.
You can identify that bud, and you can make the reverse twist of that particular bud. Reverse twist means once the front side, once the reverse side, then it will cut. Actually, the calyx portion will cut. Complete cut will be there. Then slowly just drag it from the top. The style will not be damaged. Stigma will remain intact. There is no problem, but only the other portion will come out, calyx tube as well as the petal portion along with the other male parts will come out, and then you can actually tag it with the thread that it is emasculated, and next day you can actually pollinate it. And now here the problem is actually for seed setting, you must have a proper humidity because the pollen actually requires the moisture conditions.
Yes.
The better moisture is there, the pollen will be sticky and fertilization will take place properly. Otherwise, if it is very dry, you may not get the crossed seed. That is the problem. Otherwise, this hybridization is quite simple in groundnut, but unfortunately, per cross you get only 1 or 2 seeds. That is the problem. That is inherent problem. We cannot solve that. But by practice, you can actually use this technique. It is very fast. You can do it very quickly, and you can train the skilled workers in that. In our case, the ladies here, the skilled worker, even they are not highly skilled, but still they can do it. That is possible. And one more thing I actually forgot to while delivering my lecture. Once you make the cross and grow them, how to identify the F1s actually, that is a very tricky thing.
Yes.
Now, on either side of the particular cross, you must grow female parent as well as male parent. Okay.
Okay.
Now, you know the female parent characters perfectly, how they are. And if any plant, F1 plant is exactly matching female, then definitely it is a self. That means, now another thing, while crossing actually, the flowers that are produced before you start crossing should be perfectly removed. Now, even if you leave 1 or 2, those could be selfed ones. And even after completion of your hybridization work, later also some flowers will come. You have to completely remove them daily. Then only you will retain only the crossed seeds. Otherwise, that is difficult. And another thing, while identifying the F1s, you should look for the male characters in the F1. At least some male characters will be there in F1. You can identify based on that and tag which are the true crosses and remove those which are self ones. That way you can actually identify. It is quite possible.
Okay, sir, for nice explanation, sir. But one doubt is, sir, to remove all flowers with that female plants is a task, sir, actually.
No, you can assign a labor for that. It has to be done. Otherwise, your whole process of, if you're not doing that, if the
Yes, sir.
self seeds are there, you will be playing only with the female plant. No, there will be no variation at all. That will
Sir,
be.
actually, we did the same, but it is very means. Otherwise, we can take the number of plants some few only. We can reduce the number of plants. That is good if we do not have enough labor or everything.
No, actually, as I said in my lecture, unless you have the large F2 population.
No, sir. For that note, I'm when we are going for crossing, I am talking about the female plants.
Yeah. Female plants, you can restrict the number of plants, means you will get less number of hybridized seeds. No, you cannot grow like other crops. You cannot grow large female plants and then attempt crossing. That is not possible. Either you have to grow limited number of female plants in pots or directly on the bed and then attempt the crosses.
Okay, sir. Thank you so much, sir. Thank you so much, sir.
Thank you, sir. And if any other questions from the participant? Okay. Sir, I think we have come to the end of today's presentation, and I thank profusely Dr. Nadaf, sir, for accepting our invitation at short notice and preparing a very good presentation, sir. I appreciate you. And actually, this is what our directors have wanted.
Yeah.
Like the skill sets, what you call the tips. To become a good good observer and to understand plant, how it responds, what are the seasonal variations, and how to make best use of that, whatever the climate throws at us, whatever the conditions that we face, and to make good use of that. So that's what we are actually looking for, and we actually, your presentation fitted well to whatever the requirements we had. And thank you for understanding our need and making a very good presentation, sir. Our AICRP scientists, our Institute IIGR scientists, and the students who have joined from different universities and from across from Africa also. I see some of the participants. So I thank you for making such good presentation and interacting with us. And I thank the US Dharwad for organizing this one, sir, from facilitating your presentation from their end. And I thank, sir, Olam, sir, do you have any question? Emmanuel?
Yeah.